type 3 hypersensitivity and remember, I will make it very easy. Okay, see, think there is an antigen like bacteria, microbes or any other microbes had entered the body. What happens? Then immune system get activated and produces antibodies. Okay, this antigen and antibody form antigen antibody complex. Okay, there is an antigen in the body which had come from the external, and then antibodies are produced, and those antigen and antibody get combined and produce an antigen antibody complex. Okay, what happens? These antigen antibody complexes are cleared by some immune cells like neutrophils or macrophages. They will clear this. So सब कुछ ठीक है, means this bacteria is cleared. But if these antigens are more in quantity, this antigen antibody complexes are formed in more in quantity, so the immune system might not clear them properly. Means there may be some remaining antigen antibody complexes may be there. So what these remaining antigen antibody complexes do? These antigen antibody complexes will deposit it where the blood get filtered more. Where the blood get filtered more? In the kidneys, in the skin, in the joints for synovial fluid, the joints. Okay, it got deposited here. So what happens after get deposited here? See, think this is an immune complex. It had deposited somewhere. Okay, let's take joints. It had deposited in the joints. After it deposited in the joint, see, I told in the previous lecture that these complexes are coated with some antibodies, that is IgG or IgM, right? So okay, from not previous class, from here also we can know that antibodies are IgM or IgG, right? So this immune complex contain this IgM, right? So this immune complex has IgG or IgM. So the random proteins in the blood which are circulating, which aid the destroying of these microbes in the immune complex, are complement proteins. In the blood there are some random proteins which are known as complement proteins, which help in killing of the bacteria or any foreign particle. Okay, which help the immune system. Okay, these are complement proteins. Okay, these complement proteins detect this IgG, so the complement system will activate. So first C1, C4, C3, C3A, C3B. In the previous lecture of type 2 hypersensitivity, I had told all this cascade. You can see that if you don't understand here. C3B, C5, C5A, C5B, and then from this, okay, till here only, those all will activate in another pathway, that is in the type 2. Okay, this C3A and C5A, our point of talking is here C5A and C3A. Okay, after this complement cascade had activated, due to it, because why they got activated? Because they recognized IgG. Okay, after this complement system activated and produced here C5A and C3A. How? First C1 got activated, then C4 got activated. Okay, then C4 got activated, then C3 got activated. C3 had split into C3B and C3A, and then C5 formed. Then C5 also splitted into C5A and C5B. Then our point of interest is C5A and C3A. What this C3A does? This C3A causes inflammation by calling this C3A calls the inflammatory mediators and causes inflammation. Okay, what this C5A does? See, See, this C5a calls neutrophils. Neutrophil by इधर कुछ antigen है, you come here and kill it. Okay, so the neutrophil comes and tries to take out the immune complexes which are, we are discussing about joints, right? This is a joint and these immune complexes are deposited. So this neutrophil tries to take them out by engulfing, but they won't come out. Why they won't come out? Because they are deposited deeper into the tissues, so they don't come out. So what happens for this neutrophil? It gets frustrated very much and produces some enzymes, that is proteases, reactive oxygen species, and also liposomal enzymes. Okay, it got frustrated and released proteases, reactive oxygen species, and liposomal enzymes. So what this do? Along with damaging these immune complexes which are deposited, it also damages the local tissue. It also damages the local tissue, also get damaged because of these proteins. So we can see collateral damage, that is damage to our own tissues, and also the bacteria can be seen in this type 3. Okay, we had seen collateral damage. So the tissue got damaged and we got inflammation. This is what happens in type 3. Okay, let's take some of common and most important examples of this type 3, that is systemic lupus erythematosus and post-streptococcal glomerulonephritis, then Arthur's reaction, and one more, serum sickness. Okay, we'll discuss about this in detail. First we'll go with systemic lupus erythematosus. Okay, let me take another pen. Now we are discussing about systemic lupus erythematosus from type 3 hypersensitivity. Type 3 hypersensitivity. See, in the cell there is DNA. This DNA has our double stranded DNA, right? Yes. There is dsDNA. There is Smith antigen. Smith protein, dsDNA. There are histones. I think you had seen dsDNA and histones. You are new to Smith antigen. Okay, this is also present. Smith protein. Okay, then nuclear proteins, ribonuclear proteins. Okay, sorry, ribonuclear proteins. Okay, what happens after this cell got lysed due to apoptosis, necrosis, or any type of cell death? It got outside, the DNA had exposed outside, and what happens in normal conditions? The macrophages or neutrophils engulf this DNA material, and they will the next continues very properly. But our point of interest is pathology, right? We are not interested in normal things, we are interested in abnormal things. Okay, what happens? This exposed DNA is thought by the immunity like antigen as non-self. So what it does, it produces all antibodies to all this. So you can tell in order: anti-dsDNA, anti-Smith antigen, anti-histones, anti-ribonuclear proteins. antibodies against dsDNA, Smith antigen, histones, nuclear proteins. Those are anti-dsDNA antibodies. Those are anti-Smith antigen antibodies, anti-histone antibodies, anti-nuclear protein antibodies. What these antibodies do? These antibodies, if cleared by the cell immune system, okay, but in the type 3 they will not get what? They will not get cleared by the immune system. So what happen? They go and deposit in the joints, in the skin, anywhere where the blood is filtered more, they will get accumulated there. In the skin, in the kidney, okay, they will get accumulated in these places. One more, in the pleura, in the pericardium, in the blood vessels. Okay, I told, right, after this immune complexes got deposited, there will be C3A and C3B. This C3A calls the inflammatory mediators, and this C3B calls, C5B, C5A, C3 and C5A. This C5A calls neutrophils. This C5A calls neutrophils, and these neutrophils try to engulf, but they can't engulf. So what they produce? Proteases. And what are the other? Proteases, reactive oxygen species. Then what more? Proteases, reactive oxygen species. One more, liposomal proteins. What these do? They destroy the normal tissue and also the antigen which is present. So that is collateral damage, right? So in the joints, in the SLE, due to all these enzymes and due to deposition, there will be arthritis. Okay, in the skin, there will be butterfly shaped rash. Butterfly rash or malar rash. Okay, butterfly shaped rash will be there on the nose and cheeks. There will be butterfly shaped rashes. Why rashes are coming? Because there is inflammatory mediation by C3A, right? So there is rash. Okay, we got the mechanism behind rash, and we got the mechanism behind arthritis. And in the kidney, the glomerular membrane will be damaged, so there will be proteinuria, means the protein will be leaking out due to damage of glomerular membrane. So proteinuria. Then what? Hematuria will be there. Okay, these are the things in the kidney. This is also called lupus nephritis, because proteinuria happening and hematuria is happening in SLE, lupus erythematosus, so lupus nephritis. Then this is in the kidney. In the pleura, if they got deposited, it causes pleuritis. If the pericardium, it causes pericarditis. Then in the blood vessels, it causes inflammation, right? Then it causes vasculitis. Easy, right? In the blood, vasculitis, pericarditis, pleuritis. In the kidney, it causes lupus nephritis due to it damages the glomerular filtration membrane, proteinuria and hematuria. And in the joints, it causes arthritis. In the skin, it causes butterfly shaped rash. Easy, right? Then we'll discuss, okay, this is systemic lupus erythematosus. Okay, see, in this we'll also discuss about lab diagnosis. See, it is very easy. From the pathogenesis we had discussed, we can also write lab diagnosis. In the lab diagnosis, see what I had told, the antibodies are produced against what? Produced against the DNA material, right? So anti-nuclear antibodies. Okay, then all anti-nuclear antibodies we can see. And in this, one more concept is there will be C3 complement protein and C4 complement protein reduced in the blood. Why? Because see, there is this immune complex, right? Antigen antibody complex. This antigen antibody complex consumes C3 and C4. It takes C3 and C4 protein, so it will be less in blood. And one more we can see, that is lupus erythematosus cell, LE cell. This is also called LE cell, lupus erythematosus cells. What is this lupus erythematosus cells is, there is a neutrophil which was called by C5a. This C5a called neutrophil, it engulfs the antibody coated DNA. The DNA is coated by antibody. This is antibody and this is DNA, right? In the blood. Okay, normally see, don't see this, in this DNA, this is coated by antibody. This is IgG or IgM. This is engulfed by the neutrophil or macrophage or neutrophil. Okay, this is the engulfed cell, not engulfed cell, the cell which engulfed. So this is called LE cell. The LE cell is nothing but the neutrophil or a macrophage which engulfed the antibody coated DNA. That is LE cell. And what we can see butterfly rash over cheek or nose. And what else we can see? Lupus nephritis we can see, pericarditis we can see, all those we can see. And these are mostly the lab diagnosis we can do. One more I forgot to tell, that is, see, in the type 2 hypersensitivity, type 2, there is deposition in linear pattern. That means antibodies if we do immunofluorescence, if we do, the deposition of antibodies will be in a linear pattern like this. Linearly they will get deposited. But here there will be granular pattern. That means complexes are formed irregularly. Granular pattern in what? Here type 3. In the type 3 we see granular pattern of deposition of immune complexes. In the type 1 we see the antibodies are deposited how? In linear pattern. Okay, linear pattern we see in immunofluorescence of type 2, and type 3 we can see granular pattern. Okay, that is what I forgot to tell. I'm thinking I forgot something, but I got remembered now. Okay, now we'll discuss about PSGN, Arthur's reaction, and serum sickness. See, the process is same everywhere. The immune complexes get accumulated, complement system get activated, inflammation, same thing. But here after streptococcal infection, there will be antibody. Then I'm not writing everything. There will be antigen antibody complexes. These complexes will get deposited in the glomeruli. Okay, I will write glomeruli. Then they will activate complement system get activated. Then what happen? There will be inflammation and damage, collateral damage, right? And damage. So if it is happening in the glomerulus, what we can see? We can see proteinuria here also. Proteinuria we can see, hematuria we can see. Okay, This all lead to post-streptococcal glomerulonephritis, that is proteinuria, proteins are lost, blood is also lost. Okay, then we'll go with serum sickness. See, like antivenoms, we take antivenom from horses, right? That means we inject the antigens into the horse body and we take the horse serum, and whenever a snake bites us, we inject this serum, sorry, not venom, we inject this serum, right? Why? Because this serum has anti-venom properties. The antibodies are against this venom. The antibodies are against the venom, right? So we take this serum which is produced in other animals, example like horse, and we treat ourselves when we are bitten with snake. But see, suppose this is a serum we are taking from horse and this is our body. This body has that snake poison material. So after the serum we are taking from the horse and we are injected into our body, these antibodies which are against this snake antigens, they will be killed. They will be killed. But along with the serum which is against antibodies, against antigen, against snake antigen, there are antibodies, right? Against a snake antigen, there are antibodies. These antibodies will destroy this antigen in our body. But we are not we are not taking the antibodies. From that, other proteins also will come from the serum. These other proteins act as antigen in our body and they produce IgG and IgM and will accumulate and continue the type 3 hypersensitivity. This is our point of interest. See, we are bitten by a snake and we are taking serum from other animal which historically we had taken. In the past days, the serum is taken and we are injecting to the human body. So antibodies which are present in the serum of the animal will kill the antigens against antigens of snake, and they will neutralize the poison. But there are also other proteins in the serum. They will activate our immune system and produce immune complexes. Now your doubt may be, why this foreign antibodies will not induce immune response? These antibodies also will induce immune response, but see, these antibodies and these antigens, snake venom, get neutralized because we already have venom. These antibodies we are giving, so they both get neutralized. But what can't be neutralized? The other proteins which are present with the antibodies. So those take out the type 3 hypersensitivity by forming immune complexes and they will be deposited. That is serum sickness. And one more, the Arthur's reaction. See, this is the skin and we are injecting some antigen. So what happens? The body develops antibodies. The body develops antibodies and the disease is cured. But after some years, after some days after the disease is cured, we are again injecting the same antigen only in a particular area of the skin. In the particular area of the skin, what happens? As antibodies against this antigen are already present in the body, there will be local rash or inflammation. As we are injecting into skin, there will be local inflammation or rash because antibodies are already present. So this is Arthur's reaction. Why this rash is formed? Because antibodies are already there. So they had got here and they form antigen antibody complexes and C3 get activated. This activated C3A will form what they will call inflammatory response. So inflammation is there and rash is produced. But what is the difference between this and type 4 hypersensitivity. That is, see, now there will, this is the single confusion. See, this all process will happen in 4 to 12 hours. Okay, the example test is diphtheria toxoid. If previously diphtheria toxoid has had in the body or not, we can know by this type 3 hypersensitivity mechanism. That is Arthur's reaction. Example of Arthur's reaction is diphtheria toxoid test. It will happen in 4 to 12 hours. But you all know about Montauk's test, right? In Montauk's test, what is the difference between Montauk's test and this diphtheria toxoid test? Means there also we can see inflammation and rash. Here also we can see inflammation and rash. But what is the difference? Why this Arthur's reaction is not in type 4, or why that Montauk's test is not here in this type 3? See, in the Montauk's test, the formation of rash will be in 24 to 48 hours. The time difference is there, and this is due to T cell activation, and here the previously formed antibodies will create this rash. And in the Montauk's test, which is in the type 4, it will take 24 to 48 hours. And this diphtheria toxoid test in the Arthur's reaction, it will take only 4 to 12 hours. Okay, this is about the type 3 hypersensitivity reaction, and I try to cover everything. So if you want to know more about this topic, pin in comment section, and I will try to explain those topics also. Okay, thank you. Hope you are enjoying my classes.